The inner ear, internal ear, oris interna, is the innermost part of the vertebrate ear. In vertebrates, the inner ear is mainly responsible for sound detection and balance. In mammals, it consists of the bony labyrinth, a hollow cavity in the temporal bone of the skull with a system of passages comprising two main functional parts. The cochlea, dedicated to hearing, converting sound pressure patterns from the outer ear into electrochemical impulses which are passed on to the brain via the auditory nerve. The vestibular system, dedicated to balance. The inner ear is found in all vertebrates, with substantial variations in form and function. The inner ear is innervated by the eighth cranial nerve in all vertebrates. Structure. The labyrinth can be divided by layer or by region. Bony versus membranous. The bony labyrinth, or osseous labyrinth, is the network of passages with bony walls lined with periosteum. The membranous labyrinth runs inside of the bony labyrinth. There is a layer of perilymph fluid between them. The three parts of the bony labyrinth are the vestibule of the ear, the semicircular canals, and the cochlea. Vestibular versus cochlear. In the middle ear, the energy of pressure waves is translated into mechanical vibrations by the three auditory ossicles. Pressure waves move the tympanic membrane, which in turn moves the malleus, the first bone of the middle ear. The malleus articulates to incus, which connects to the stapes. The footplate of the stapes connects to the oval window, the beginning of the inner ear. When the stapes presses on the oval window, it causes the perilymph, the liquid of the inner ear, to move. The middle ear thus serves to convert the energy from sound pressure waves to a force upon the perilymph of the inner ear. The oval window has only approximately 1/18 the area of the tympanic membrane and thus produces a higher pressure. The cochlea propagates these mechanical signals as waves in the fluid and membranes, and then converts them to nerve impulses which are transmitted to the brain. The vestibular system is the region of the inner ear where the semicircular canals converge, close to the cochlea. The vestibular system works with the visual system to keep objects in view when the head is moved. Joint and muscle receptors are also important in maintaining balance. The brain receives, interprets, and processes the information from all these systems to create the sensation of balance. The vestibular system of the inner ear is responsible for the sensations of balance and motion. It uses the same kinds of fluids and detection cells, hair cells, as the cochlea uses, and sends information to the brain about the attitude, rotation, and linear motion of the head. The type of motion or attitude detected by a hair cell depends on its associated mechanical structures, such as the curved tube of a semicircular canal or the calcium carbonate crystals, otolith, of the saccule and utricle. Development. The human inner ear develops during week 4 of embryonic development from the auditory placode, a thickening of the ectoderm which gives rise to the bipolar neurons of the cochlear and vestibular ganglions. As the auditory placode invaginates towards the embryonic mesoderm, it forms the auditory vesicle or otocyst. The auditory vesicle will give rise to the utricular and saccular components of the membranous labyrinth. They contain the sensory hair cells and otoliths of the macula of utricle and of the saccule, respectively, which respond to linear acceleration and the force of gravity. The utricular division of the auditory vesicle also responds to angular acceleration, as well as the endolymphatic sac and duct that connect the saccule and utricle. Beginning in the 5th week of development, the auditory vesicle also gives rise to the cochlear duct, which contains the spiral organ of Corti and the endolymph that accumulates in the membranous labyrinth. The vestibular wall will separate the cochlear duct from the perilymphatic scala vestibuli, a cavity inside the cochlea. The basilar membrane separates the cochlear duct from the scala tympani, a cavity within the cochlear labyrinth. The lateral wall of the cochlear duct is formed by the spiral ligament and the stria vascularis, which produces the endolymph. The hair cells develop from the lateral and medial ridges of the cochlear duct, which together with the tectorial membrane make up the organ of Corti. Histology. Organ of Corti. Rosenthal's canal or the spiral canal of the cochlea is a section of the bony labyrinth of the inner ear that is approximately 30 mm long and makes 2 3/4 turns about the modiolus. There are several specialized types of cell in the inner ear. Among these are hair cells, pillar cells, Bircher's cells, Claudius cells, and Dieter's cells, phalangeal cells. The hair cells are the primary auditory receptor cells and they are also known as auditory sensory cells, acoustic hair cells, auditory cells, or cells of Corti. The organ of Corti is lined with a single row of inner hair cells and 3 rows of outer hair cells. The hair cells have a hair bundle at the apical surface of the cell. The hair bundle consists of an array of actin-based stereocilia. Each stereocilium inserts as a rootlet into a dense filamentous actin mesh known as the cuticular plate. Disruption of these bundles results in hearing impairments and balance defects. Pillar cells are found in the organ of Corti and act as supporting cells for hair cells. They are divided into 2 types, inner and outer. Outer pillar cells are unique in that they are freestanding cells with no contact to adjacent cells except at the bases and apices. Both types of pillar cell are characterized by the presence of thousands of cross-linked microtubules and actin filaments in parallel orientation. They provide mechanical coupling between the basement membrane and the mechanoreceptors on the hair cells. Bircher's cells are found in the organ of Corti where they are present only in the lower turn of the cochlea. They lie on the basilar membrane beneath Claudius cells and are organized in rows with the number of rows the number of which varies between species. The cells interdigitate with each other, and project microvilli into the intercellular space. They are supporting cells for the auditory hair cells in the organ of Corti. They are named after German pathologist Arthur Bocher, 1831–1889. Claudius cells are found in the organ of Corti located above rows of Bircher's cells. Like Bircher's cells, they are considered supporting cells for the auditory hair cells in the organ of Corti. They contain a variety of aquaporin water channels and appear to be involved in ion transport. They also play a role in sealing off endolymphatic spaces. They are named after the German anatomist Friedrich Matthias Claudius, 1822–1869. Dieter's cells, phalangeal cells, are a type of neuroglial cell found in the organ of Corti and organized in 1 row of inner phalangeal cells and 3 rows of outer phalangeal cells. They are the supporting cells of the hair cell area within the cochlea. They are named after the German pathologist Otto Dieters, 1834–1863, who described them. Hensen's cells are high columnar cells that are directly adjacent to the 3rd row of Dieter's cells. Hensen's stripe is the section of the tectorial membrane above the inner hair cell. Newell's spaces refer to the fluid-filled spaces between the outer pillar cells and adjacent hair cells and also the spaces between the outer hair cells. Hardisty's membrane is the layer of the tectoria closest to the reticular lamina and overlying the outer hair cell region. Reisner's membrane is composed of 2 cell layers and separates the scala media from the scala vestibule. Huschke's teeth are the tooth-shaped ridges on the spiral limbus that are in contact with the tectoria and separated by interdental cells. Physiology. Neurons within the ear respond to simple tones, and the brain serves to process other increasingly complex sounds. An average adult is typically able to detect sounds ranging between 20 and 20,000 Hz. The ability to detect higher pitch sounds decreases in older humans. The human ear has evolved with 2 basic tools to encode sound waves, each are separate in detecting high and low frequency sounds. George von Bakaschi, 1899–1972, employed the use of a microscope in order to examine the basilar membrane located within the inner ear of cadavers. He found that movement of the basilar membrane resembles that of a traveling wave, the shape of which varies based on the frequency of the pitch. In low-frequency sounds, the tip, apex, of the membrane moves the most, while in high-frequency sounds, the base of the membrane moves most. Clinical significance. Interference with or infection of the labyrinth can result in a syndrome of ailments called labyrinthitis. The symptoms of labyrinthitis include temporary nausea, disorientation, vertigo, and dizziness. Labyrinthitis can be caused by viral infections, bacterial infections, or physical blockage of the inner ear. Another condition has come to be known as autoimmune inner ear disease, AIED. It is characterized by idiopathic, rapidly progressive, bilateral sensorineural hearing loss. It is a fairly rare disorder, while at the same time, a lack of proper diagnostic testing has meant that its precise incidence cannot be determined. Comparative anatomy. Birds have an auditory system similar to that of mammals, including a cochlea. Reptiles, amphibians, and fish do not have cochleas but hear with simpler auditory organs or vestibular organs, which generally detect lower frequency sounds than the cochlea. The Cochlear System. In reptiles, sound is transmitted to the inner ear by the stapes, stirrup, bone of the middle ear. This is pressed against the oval window, a membrane-covered opening on the surface of the vestibule. From here, sound waves are conducted through a short perilymphatic duct to a second opening, the round window, which equalizes pressure, allowing the incompressible fluid to move freely. Running parallel with the perilymphatic duct is a separate blind-ending duct, the lagina, filled with endolymph. The lagina is separated from the perilymphatic duct by a basilar membrane, and contains the sensory hair cells that finally translate the vibrations in the fluid into nerve signals. It is attached at one end to the saccule. In most reptiles, the perilymphatic duct and lagina are relatively short, and the sensory cells are confined to a small basilar papilla lying between them. However, in birds, mammals, and crocodilians, these structures become much larger and somewhat more complicated. In birds, crocodilians, and monotremes, the ducts are simply extended, together forming an elongated, more or less straight, tube. The endolymphatic duct is wrapped in a simple loop around the lagina, with the basilar membrane lying along one side. The first half of the duct is now referred to as the scala vestibuli, while the second half, which includes the basilar membrane, is called the scala tympani. As a result of this increase in length, the basilar membrane and papilla are both extended, with the latter developing into the organ of Corti, while the lagina is now called the cochlear duct. All of these structures together constitute the cochlea. In mammals, other than monotremes, the cochlea is extended still further, becoming a coiled structure in order to accommodate its length within the head. The organ of Corti also has a more complex structure in mammals than it does in other amniotes. The arrangement of the inner ear in living amphibians is, in most respects, similar to that of reptiles. However, they often lack a basilar papilla, having instead an entirely separate set of sensory cells at the upper edge of the saccule, referred to as the papilla amphibiorum, which appear to have the same function. Although many fish are capable of hearing, the lagina is, at best, a short diverticulum of the saccule, and appears to have no role in sensation of sound. Various clusters of hair cells within the inner ear may instead be responsible. For example, bony fish contain a sensory cluster called the macula neglecta in the utricle that may have this function. Although fish have neither an outer nor a middle ear, sound may still be transmitted to the inner ear through the bones of the skull, or by the swim bladder, parts of which often lie close by in the body. The vestibular system. By comparison with the cochlear system, the vestibular system varies relatively little between the various groups of jawed vertebrates. The central part of the system consists of two chambers, the saccule and utricle, each of which includes one or two small clusters of sensory hair cells. All jawed vertebrates also possess 3 semicircular canals arising from the utricle, each with an ampulla containing sensory cells at one end. An endolymphatic duct runs from the saccule up through the head, and ending close to the brain. In cartilaginous fish, this duct actually opens onto the top of the head, and in some teleosts, it is simply blind ending. In all other species, however, it ends in an endolymphatic sac. In many reptiles, fish, and amphibians this sac may reach considerable size. In amphibians the sacs from either side may fuse into a single structure, which often extends down the length of the body, parallel with the spinal canal. The primitive lampreys and hagfish, however, have a simpler system. The inner ear in these species consists of a single vestibular chamber, although in lampreys, this is associated with a series of sacs lined by cilia. Lampreys have only 2 semicircular canals, with the horizontal canal being absent, while hagfish have only a single, vertical, canal. Equilibrium. The inner ear is primarily responsible for balance, equilibrium, and orientation in 3-dimensional space. The inner ear can detect both static and dynamic equilibrium. 3 semicircular ducts and 2 chambers, which contain the saccule and utricle, enable the body to detect any deviation from equilibrium. The macula sacculi detects vertical acceleration while the macula utriculi is responsible for horizontal acceleration. These microscopic structures possess stereocilia and 1 kinocilium which are located within the gelatinous otolithic membrane. The membrane is further weighted with otoliths. Movement of the stereocilia and kinocilium enable the hair cells of the saccula and utricle to detect motion. The semicircular ducts are responsible for detecting rotational movement.
Please subscribe and thanks for watching.